This is a production of Cornell University.
Thank you, Johannes. I'm excited to share with you all today my work on modeling microbial community control on soil organic carbon persistence. Soil organic carbon persistence is important for us since it brings multiple benefits for our agricultural production, for soil health, soil biodiversity, and may help us mitigate climate change. We know that soil organic carbon persistence is an ecosystem property which deals with the dynamic balance of carbon incoming to the soil, primarily from plant material, and carbon leaving the soil, primarily with microbial respiration. And soil organic carbon persistence basically is controlled by many environmental and biological factors. The factors that are well known and including that in most carbon models are organomineral interactions, soil aggregation, soil temperature, and soil moisture. However, now we talk more and more about the emerging factors such as molecular diversity, microbial carbon use efficiency, spatial heterogeneity, temporal variability, which all deal mainly with the functioning of soil microbial community and of its immediate soil environment. However, the role of microbial community composition in the soil organic carbon persistence is still largely unknown. Why is it important for us to understand it better? On the one hand, it may help us improve our global and regional carbon cycling models. But even more importantly, it is essential for us to improve our understanding of soil microbial community for informed decisions about our human interventions and soil management practices, because they may influence greatly soil microbial community composition and can have specific repercussions for our carbon accrual. So, in my dissertation, I am addressing the question of soil organic carbon persistence from three different perspectives. In my first chapter, I deal mainly with global carbon models, and I'm looking at what locations soil organic carbon persistence is the main source of global land carbon cycling uncertainty. In chapter 2, I delve more into mechanisms of carbon cycling, and I'm interested in knowing whether microbial community composition controls soil organic carbon persistence. And in my chapter 3, I look at human interventions, in particular at enhanced rock weathering. And my question is whether it increases or decreases soil organic carbon persistence. My first chapter deals with the fact that there are many global land carbon cycling models. And according to the Global Carbon Budget Project, on average, these models predict that there is a growing land carbon sink that is supposed to help us to mitigate climate change. However, when we look at individual predictions of these global models, we see that the uncertainty is quite high and their predictions deviate quite a lot, not only in the magnitude of the predicted land carbon sink, but even whether it is actually a sink or a source. And my question is, what is the main source of this uncertainty? So the objectives of first chapter are to determine the main source of uncertainty in the global land carbon estimations, whether it is soil carbon persistence or vegetation turnover or net primary production. I would assess these prevalent sources of uncertainty geographically, and then I would find out whether, under what geographical conditions, the relative uncertainty of soil carbon persistence is higher. And my hypothesis is that soil carbon persistence is the primary source of uncertainty in the global land carbon predictions. How would I do that? I'm working with the output of the 16 global land carbon models from 29 Model Intercomparison Project. I take their global outputs, and for each pixel, I estimate uncertainty of their predictions as mean average deviation. Then I assume that I can estimate the quasi-steady state ecosystem carbon as a product of carbon input, NPP, and carbon turnover consisting of vegetation and soil turnover time. In this sense, a soil turnover time would be my proxy for a soil carbon persistence. And once I do that, I can propagate uncertainty from different components to the uncertainty of the total ecosystem carbon. And once I do it for every pixel, I can get the global weighted average and a special distribution of different uncertainty components. What I'm seeing so far is that my hypothesis is actually not supported. And I see that globally, NPP and soil carbon persistence contribute almost equally to the general uncertainty of the land carbon predictions, NPP being slightly ahead. And when we look at the geographic distributions of the primary uncertainty sources, then the interesting thing is that soil carbon persistence is the main source of uncertainty in the north, where we have large permafrost carbon stock, and also in the temperate and subtropical croplands. Now, this is quite exciting to know because these areas are crucial for our global food production, and the deviations between model predictions show us that we are quite unsure how soil carbon stocks develop in these agricultural lands in the context of both climate change and land use change and agricultural production. And the reason for that could be that we actually lack a lot in understanding of soil carbon processes and how our human interventions influence them. And as a result, we have rather different and rather simplistic estimations of these effects in global models. So I hope that we will gain more information on carbon processes that will inform our models in the future. And in my second chapter, I specifically deal with something that is not included in most global carbon models, namely microbial community composition. So all the models we talked about in chapter 1 are first order models that do not have any microbial biomass pools in their structure. Instead, they represent carbon turnover in soils as a movement from faster to slower cycling conceptual pools. More modern microbial models have a carbon pool in their structure, but usually no information on microbial community composition. Usually it's just one single microbial carbon pool. And very few models do have certain distinction between different types of microbes in their structure. And the example shown here is a mimics model that has different pools for copiotrophic and oligotrophic microbes. But in general, we see that for now we lack the understanding of microbial community features that are important for carbon cycling to adequately include community information in carbon models. On the other hand, several recent studies showed that microbial carbon use efficiency is a key parameter for predicting soil organic carbon persistence. And I'm interested in this parameter as something that is very likely to be affected by the microbial community composition. So my objectives and hypotheses for this chapter are to determine whether soil microbial community composition controls soil organic carbon persistence, to find specific features of soil microbiome that are predictive of soil organic carbon persistence, and to establish whether environment or microbial community has a stronger effect on soil organic carbon persistence. My hypothesis is that microbial community composition has a stronger direct effect on carbon persistence than the environment. And to answer these questions, I'm taking a fully data-driven approach. I'm not using any process-based model with a pre-developed structure. I am not using any prior assumptions about variable importances. Instead, I'm analyzing the data using machine learning techniques to let the
No, it's okay.
structure
I just
and the
want to try.
relationships between variables emerge from the
No,
data
no, no,
itself.
no.
For that, I'm
Your business
using
is much more important at that moment.
a large, excuse me. For that, I'm using a large observational data set across Europe from the Lucas Soil Survey with 885 sites, where I have climate data, soil properties, potential soil respiration data, and microbial sequencing data, both for bacteria and for fungi. Unfortunately, however, the observational data set doesn't have the measured values for microbial carbon use efficiency, which is quite difficult to measure. But instead, I'm using a respiratory quotient or a ratio between microbial respiration and microbial biomass as a proxy for carbon use efficiency. And in most models that are in use now, carbon use efficiency is very strongly inversely related to the steady state respiratory quotient, as shown here on the example of the millennial model. Now, in reality, this relationship would not be that strong because in reality, microbes do have more processes of how they use carbon as opposed to how it's represented in the models. So in the models, we only have carbon growth or microbial growth and microbial respiration as the two possible uses of carbon, while in reality, of course, carbon is also used for production of extracellular enzymes and other exudates. So we keep in mind that this relationship is so strong only for the models, but for the modeling perspective, respiratory quotient is quite a good proxy. So, since I have a very large number of potential predictors, both environmental and microbial, I'm using a machine learning framework to figure out which predictors are most important. So I start from fitting an ensemble of random forest models. I use then Shapley values to rank variable importances and a forward selection scheme to create a cutoff for the optimal number of variables used in the model. Once the important predictors are selected, I study the relationships between individual variables and I use structural equation modeling to see direct and indirect effects of my predictor variables on the respiratory quotient. What I find so far is that bacterial taxa are quite good predictors for the respiratory quotient, and they explain about 50% of its variability, which is on par with environmental variables. Functional annotation does not increase the prediction power, which is quite surprising. And also, fungal data has a lower prediction power than bacterial data in my study. But the most interesting is that when we add bacterial and environmental variables together, the increase in R square is quite small, only plus 5% compared to using either environment or bacteria alone. And that tells us that there is a lot of redundancy between microbial community variables and the environment. When we look at individual variables, then unsurprisingly, among environmental variables, the water content is the primary predictor. Well, soil moisture content's effect on respiration is quite well known and included in most contemporary models. However, then we see that there is a number of microbial orders that also have a very strong effect on the respiratory quotient. Uh, Especially, we see that Acidobacterialis and Propionibacterialis are the prominent orders affecting the overall community's respiration. On the other hand, we see that several environmental variables are very tightly aligned with microbial order abundances. In particular, pH is correlated and probably determining the abundances of many microbial taxa. And when we combine all this information in a structural equation model, we see that the direct effect on the respiratory quotient among environmental variables is the strongest by water content and clay. However, other environmental variables primarily directly affect microbial community composition. And then, in turn, microbial order abundances have a strong direct effect on respiratory quotient of their own. So we see that microbial variables do affect respiratory quotient and do control it. However, they are in turn controlled by the environment, and therefore environment's indirect effect on the respiratory quotient is major. What does it tell us about modeling? Well, basically, we have two options how we can deal with carbon use efficiency in the models. One way would be to have microbial community implied and not present in the model structure. And then we will have to see what environmental variables affect microbial communities' respiratory quotient or carbon use efficiencies, and use these environmental variables to adjust CUE parameter. Option two would be more explicit to model certain microbial community types from the environment and then use these microbial community types to adjust carbon use efficiency parameter. Finally, in my third chapter, I'm looking at enhanced rock weathering, which is a promising direct carbon removal technique that aims to spread crushed basalt on agricultural fields to induce carbonate formation. Well, there are both strong proponents and strong critics of this technique. However, most studies of it are focusing on the inorganic carbon, which is the main mechanism how it's supposed to work. I, on the other hand, am most interested in how it affects organic carbon persistence. And there are two possible pathways how it does. The first is that calcium and magnesium ions are released into the soil during enhanced rock weathering, which may induce cation bridging in the soil, which may affect organomineral association and soil aggregation. On the other hand, enhanced rock weathering is known to raise soil pH. And as we saw in the previous chapter, soil pH is very important in determining what kind of microbial community we will get in the soil, which in turn controls soil organic carbon persistence. So my objectives here are to determine whether enhanced rock weathering leads to increase or decrease of soil organic carbon over a range of soils, to model enhanced rock weathering influence on mineral association, aggregate formation, microbial carbon use efficiency, and to predict potential effects of enhanced rock weathering on soil organic carbon throughout US croplands. My hypothesis is that enhanced rock weathering improves soil organic carbon persistence in acidic soils through changing microbial carbon use efficiency. And for this task, I'm taking a collaborative approach, bridging experiment and modeling. I'm going to use soil samples collected by our former lab member, Rachel Davenport, throughout the United States. And I will collaborate with my colleague from Lehmann Lab, Maria Rivera, who will do mostly experimental work while I will do the modeling part. We're collaborating in the framework of EDF Atkinson project that we got for this study. And basically, the goal is first to use incubation experiment to measure the effects of enhanced rock weathering on different carbon pools, and then to add enhanced rock weathering component to a microbial carbon model to parameterize it with the data from the experiments, and then to apply that model to United States croplands. For the model to use, I selected the millennial soil organic carbon model, mainly because it includes all three mechanisms that are likely to be affected by enhanced rock weathering, namely microbial carbon use efficiency, the formation of mineral-associated organic matter, and the formation of soil aggregates. So my task will be to add enhanced rock weathering component to it and parameterize it in a way that it controls the three fluxes that are affected by the weathering. Finally, conclusions from my work are that microbial community controls soil organic carbon persistence, but environment control help us improve our understanding of nature, but only if it is used as a tool rather than as a replacement for such understanding with automatic prediction. And I would like to thank everyone for their attention, and I would like to thank my committee members, Dr. Johannes Lehmann, Dr. Daniel Buckley, Dr. Yin Sun. Thank you very much, Johannes, for all your support in my work and in my difficult circumstances. And I thank the Lehmann Lab Group, which is amazing and always lifts my spirits. And I would like to thank my collaborators, Dr. Emily Oldfield from EDF, Maria Rivera, Nazim from Computer Sciences Commons Lab, and Lou Lab Group, with whom I started my first chapter. And my funding comes from AI Climate Institute, EDF Atkinson grant, and I use NSF Access for Advanced Computing. Thank Thank you.
Thank you very much. Fantastic, Kostya. Thank you so much. We have a couple of minutes for Q&A, and I'll run around with this thing here so he can really hear you.
Thank you for the presentation. I was curious about what the piecewise structural equation modeling that you used. Is that taking account the nonlinear relationship between the system components? And then also, is there any like hierarchical structure that your model isn't taking into account?
Yeah, thank you. That's a very good question. So for structural equation modeling, currently we're using linear relationships, and I'm transforming the variables to basically maximize the linear component of that relationship. But at the same time, I'm collaborating with postdoc Nassim from the computer sciences, Carla Gomez's lab, where we are trying to basically use machine learning to discover nonlinear relationships between the variables and then include them in the structural equation modeling scheme. As for a hierarchical structure, I basically start from assuming that all microbial variables depend on all environmental variables, and then I sequentially remove the insignificant variables, and I come up with models where only the significant links are preserved.
Any other questions? If not, that's not the case, then very clear. Some murmurings here on the right side, Kostya. Very clear. Thank you so much for being here for Kostya, for all your support. Now this might be the model we're headed to for our seminars. Anyways, we'll be moving to the next speaker, and join me in thanking Kostya again.
This has been a production of Cornell University, on the web at cornell.edu.